So, hi, my name is Petronio Gisulgo Jr. BSME4. So, the TED Talk is all about the animal cell structure. So, what is animal cell structure? So, it is a typical of a eukaryotic cell enclosed by a plasma membrane and containing a membrane-bound nucleus and organelles, unlike the eucharistic cell of plants and fungi. Animal cells do not have cell wall. Most cells, both animal and plant, range in size between 1 and 100 micrometers and are thus visible only with the aid of a microscope. Most animal cells are diploid, meaning that there are chromosomes exist in homologous pairs. Different chromosomal ploidies are also, however, known as to occasionally occur. So the proliferation of animal cells occur in a variety of ways. In instances of sexual reproduction, the cellular process of meiosis is the first necessary so that haploid daughter cells or gametes can be produced. So two haploid cells then fuse to form a diploid zygote, which develop into a new organisms as its cells divide and multiply. The earliest fossil evidence of animals dates from Vindian period. So it is ranged to 650 to 400, 544 million years ago. So colinterate type creatures that left traces of their soft bodies in shallows water sediment. So cells were discovered in 1665 by British scientist Robert Hooke, who first observed them in his crude, by today's standard, 17th century optical microscope. In fact, Hooke coined the term cell in biological context when he described the microscopic structure of quark like a tiny bare room of Planck's cell. So the tool microscope has been fundamental tool in field of cell biology and is often used to observe living cells in culture. So it is used to determine the type of cells either for human, animal or plants. Here are some various components that are found in animal cell. First is centriolysis. It is a self-replicating organelles made up of 9 bundles of microtubules and only found in animal cells. They appear to help in organizing cell division but aren't essential to the process. So next is the Golgi or Golgi apparatus. So it is a distribution of shipping department for cells chemical products. It modifies protein and fats, build in endoplasmic reticulum and prepares them for export to outside of the cell. So mo to siya ang endoplasmic ang Golgi apparatus. So next is the nucleus. So the nucleus is highly specialized organelles that serves as the information processing and administrative center of cell. So the organelle has two major functions. It stores the cell's hereditary material or DNA and it coordinates the metabolism, protein synthesis, and reproduction or cell division. So next is the microfilament. So it is solid rods made of globular protein called actin. These filaments are primarily structural in functions and are an important component of the cytoskeleton. So also for animals, lysosomes, the main function of these microbodies is digestion. So just like human. So lysosomes break down cellular waste products and debris from outside the cell into simple compounds which are transferred into cytoplasm as new building materials. So just like the human ways of ang digestion bitaw, parehara po siya sa animals. So animal cells also have mitochondria. So mitochondria are oblong-shaped organelles that are found in cytoplasm of every eukaryotic cell. In the animal cell, that they are main power generators, converting oxygen and nutrients into energy. So just like the human being, so we eat food just to convert into energy. So mo po gyapon siya ang sa animals. So endoplasmic reticulum, so the endoplasmic reticulum is a network of sacs manufactures transport chemical compounds for use inside and outside of the cell. It connected to the double layer nuclear envelope, providing a pipeline between nucleus and cytoplasm. And next is the microtubules. So these straight hollow cylinders are found throughout the cytoplasm of all eukaryotic cells or prokaryotes. Prokaryotes don't have them. So and carry out a variety of functions ranging from transport to structural support. So mao to siya ang sa tang microtubules. So next is the peroxisomes. So microbodies are diverse group of organelles that are found in the cytoplasm, roughly spherical and bound by a single membrane. There are several types of microbodies, but peroxisomes are most common. So next is the plasma membrane. All living cells have a plasma membrane that encloses their contents in prokaryotes. So the membrane is inner layer of protection surrounded by a rigid cell wall. So eukaryotic cell animals have only the membrane to contain and protect their contents. So these membranes also regulate the passage of molecules in and out of the cells. So that's the work of plasma membrane for animals or animal cell. So that's its rule. So next is the ribosomes. So all living cells contains ribosomes. Tiny organelles composed of approximately 60% RNA and 40% protein. In eukaryotes, ribosomes are made of 4 strands of RNA. And in prokaryotes, they consist of 3 strands of RNA. So they are different. And according to their kanang counting sa ilahang strands. So muna siyang inyong makita sa video ang picture sa animal cells. Muna siya tanan. So again, animal cell TED Talk. Thank you, sir.